Thank you, Laurie, for this kind introduction. Thank you all. Thanks to Omar and Doug for a fantastic conference. And thank you for the dinner yesterday, which we all enjoyed, including the final musical entertainment. I will introduce this day by telling about 3 discoveries I made, some very early, and I didn't understand the implications of them, but they are all related to the role of water. You can find the full story in the journal that Laurie has founded, which is called Molecular Frontiers Journal, and you can see the reference there. The inspiration of this came from a Molecular Frontiers meeting in 2009 in Bangalore, and the young people there inspired me. They were 2 busloads. This is the morning one, because you see Dick Sayre is still awake, and these are the older students, like 16 to 18. And I asked them, what do you think is the role of water for life? And they were very sophisticated. You could say that they were destroyed by the teachers and textbooks. So they gave answers like, well, water is a perfect solvent, dissolves hemoglobin. It's also a good transporting liquid, blood, for instance. Whereas in the afternoon, a younger category came, and then they generally were more prone to think out of the box than the older ones. I think this is very interesting observation. Maybe they were more brave. And a girl then said the following. She had observed the foam at the ocean beach, and we know that it's proteins and also nucleic acids and so on. And she thought that the water interface makes the molecules of life meet. And of course, we can say that they become concentrated and they react with each other. And we all know about the hydrophobic effect. We know that this is a membrane, and this is a molecular dynamic simulation of a membrane. You see the water molecules moving very fast. Albert Einstein would have loved this, showing the fast motion of the rotation being typically a picosecond of the water molecules, and the sluggish big membrane. So the water molecules makes the lipid molecules stick together. So you need the water to get the lipids to go together. So now comes my first discovery. 50 years ago, I was studying sandwich polyethylene that I stretched, and I put small molecules in this polyethylene, and I studied their orientation, and I could deduce all kinds of things. But I noticed that molecules like benzoic acid, which is either benzoate, then it's negatively charged, or even this guy, which has a dipole moment, don't want to go into non-polar solvents. And still I observed that all the benzoic acid went into the polyethylene. And the reason is that it forms a dimer. And here we have a model for how the DNA bases recognize each other by hydrogen bonds. So you see here that the dimer is, I have an extra one, a stronger one here. So these hydrogen bonds, it's a pair of hydrogen bonds that makes the 2 dipole moments exactly compensate each other. And the dimer is suddenly not a polar molecule anymore. It's rather hydrophobic, and it goes into the film. I noticed that, and I could study it, the orientation, but I didn't reflect very much about the hydrogen bonds. So this is what happens, and you could say that this looks like the DNA bases. having a hydrophobic part, and you had a pair of hydrogen bonds. This was exactly what Watson and Crick thought about when they proposed the structure of the DNA double helix. So they guessed, quite an educated guess, that guanine and cytosine and adenine and thymine formed base pairs. It was a guess. They had no evidence, and they just noticed that the percentage, the content of adenine was the same as thymine, so it was reasonable. And you may ask why they didn't understand why it was specific. Well, they got the idea from Rosalind Franklin's diffraction pattern, which told them that there was a stacking of bases. So that gave them the idea of the structure of the DNA. But the AT and GC, they thought it was 2 hydrogen bonds in both, like benzoic acid. So they looked, the guanine cytosine looked like this, and they missed the 3rd hydrogen bond, this one, which would immediately explain this specificity. But they also missed something else, much more important. And this, Laurie mentioned the discovery of an extended form of DNA, which we call sigma DNA. And we are not sure about it, but we think that it's a stacked, there are 3 bases stacked, and then it's a gap. This is just a back of the envelope calculation. I ended up with either this form or this form. In both cases, it's important to keep a number of bases stacked. So the hydrophobic interaction is still there. I will not go into detail, but at this very moment, experiments are done here at Berkeley to further study this form and try to find compelling evidence for the triplet of base stack here. Is it a coincidence that the same structure is found in a complex with homologous recombination enzymes? These are one of the oldest proteins we have on Earth, because REC-A, which is behind the mutations of bacteria, that's the most important thing for life of bacterium, to be able to get under the radar and change its structure. And in, this is prokaryotes. And eukaryotes, the enzyme is called RAD51. And that's the one that mother and father's genes are recombined in new ways. So it looks the same. It's about 50% elongated DNA, and it's found here in the crystal structure that indeed the base pairs look like this. A student of mine, Anna Raymer, based on electron microscope electron densities, modeled this, and she indeed showed that 3 bases are stacked, and then a hydrophobic group from the protein is sticking in, intercalating between the bases. So there is a relation. Again, it has to do with the DNA stacking. So we could list a number of biological roles of stretched DNA. The stacked bases in B conformation is the minimum stable construct. Perpendicular bases also provide critical base pairing in search for homology. And finally, this structure, because of the DNA is extended, is sufficiently unstable to dissociate when mission is completed. That's the REC-A dissociates. But there is another thing. How does the DNA recognize itself? Well, we see the base pairs, AT, 2 base pairs, GC, 3 base pairs. But these base pairs, they are rather weak if water is in presence. Water will compete and want to either form hydrogen donating or accepting roles. So the recognition would best work in a dry environment where we have no water. Hydrophobic effect makes DNA bases stack. So the bulk water is needed for the hydrophobic effect here, you see? And this is a little counterintuitive. The role of water is to keep the inside of the DNA dry. So the black here is the surrounding. The surrounding water is making the DNA bases stack and make it hydrophobic, and makes the hydrogen bonds exerting their full recognition power. So we can also say that this is another speculation, another direction. 3 bases, you find that in another context, that's in the codon, the RNA. So if the same principle applies to RNA, this might be the explanation why we have 3 bases in DNA. You know, the genetic code, it's 3, not 2 bases out of the 4 bases that determines which amino acid should go next into the peptide. You could say that maybe the 3 is due to the stretching here. And the textbook explanation of 3 is that, well, you have 4 bases, and if you only have the combinations of 2 bases, there would only be 16 combinations, and that's not enough. We need more than 20 different amino acids to build up a protein. And so we take the next possibility. That's 3 bases. That's 4 to the 3, that's 64. That's more than you need. So I'm bravely postulating here that this is not the origin at all. The origin is that the 3 bases is the most stable isolated configuration in the stacked nucleic acids. So if I now should summarize what I've told you, you start by creating a hydrophobic environment. And if we go back to the talks we heard yesterday, this goes into the protein folding. It also, the nucleobase stacking. And to make a precise or efficient function of the system, nucleic acid or protein, base pair recognition, that's of course very important with the fidelity in the recognition. We must not have many errors, so this is extremely important. But we also heard from Professor Gray yesterday that the efficiency of the redox system varies enormously depending on whether water is kept outside. So there are all kinds of cases where creating a hydrophobic environment is extremely important. And we may ask, has nature also exploited the possibility of using this stable triplet base stacks behind the genetic code? So I will not tell you about the last point, but that's the most recent one, and that's where we have the collaboration here with Berkeley. And that is whether there is something called hydrophobic catalysis. And you could read in this PNAS paper where one of my students, Bobo Feng, has studied how the single molecule pulling force is affected by the presence of hydrophobic agents in the water. That means in a continuum model that Ari Worschel spoke about yesterday, that if we lower the dielectric constant, and we can show that indeed we see a catalytic effect of diaglym, for instance, which is the monomer of a polyethylene glycol. And that means that this may be indeed how homologous recombination works, how the enzymes, RECA in bacteria and RAD51 in us, is catalyzing the gene exchange. So I think that's what I wanted to say in my 20 minutes. I think I was a little short here, but Are we waiting for the next speaker now? Or?
We're waiting for the next speaker who's currently en route. So we have three choices. You can answer some questions.
Okay.
We might have Professor Brewer go a little bit early, or you could keep talking. So
well, that's...
Why don't we, if there's any questions from the floor, maybe we'll take those now. I do know that our next speaker is en route, so...